Okay. Now we are live. Ah, got it.
We start recording now.
Good afternoon, everyone. Good morning, good afternoon, good evening, whatever your country is there. Uh, so it is our 6-2 webinar. Uh, just a second. I need to mute YouTube. Okay. So it's our 6-2 webinar in the series. We are very glad that today Dr. Arindam Ghatak is joined as a speaker in this webinar series. What is the reason to invite him in this platform? One is obviously is a very good scientist, young scientist, and many work he already done in this field. And another thing, he's worked on millet. So as you all know, this is the International Year of Millet 2023. So ending the 23 should be someone who is working on millet. So Arindam is a perfect for this, and we already done 3-4 webinars on millet. All are available on our YouTube channel. So I request Shoma today to introduce Bioingene a little bit, then our speaker Arindam. We will collect all your questions from YouTube chat box. We'll give you a link for certificate applications for today's webinar only in YouTube chat box during the end of this webinar. And you need to submit it. Use the same email ID which is used for the webinar registrations, because this is your registered email ID for Bioengine. And certificate will be available after 2-3 days, and we will intimate you when the certificate is uploaded on your bioingene.com website. So I request Shoma to introduce our speaker. And
Hello everyone. Welcome to another Bioengine webinar. Bioengine is a non-profit organization created to promote plant research worldwide. This is a platform from which plant scientists can present their research to the world, and we hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on building the future of plant science. We are very grateful to our guest speaker who has accepted our invitation to share his research insights with us today. We are also thankful to you, the viewers of our Bioengine family, for your interest in Bioengine webinars, your constant support and appreciation. We have received a huge response to the registration for today's webinar. You can register for more such webinars through our website, bioengine.com, and the social media pages. You are live viewing this webinar and you can watch it again on the Bioengine YouTube channel. So please remember to subscribe so that you don't miss out on any of our live. After attending today's talk, you can apply for a certificate of participation via the feedback link, which will be provided in the YouTube chat after the presentation. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is Millets for Sustainable Future, a Perspective on Abiotic Stress and BNI by Dr. Arindam Ghatak. Dr. Arindam Ghatak is a senior scientist at the Molecular Systems Biology Lab, or MOSIS, Department of Functional and Evolutionary Ecology, University of Vienna, Austria. He completed his BTech, MTech, and PhD from D.Y. Patil University, School of Biotechnology and Bioinformatics, Navi Mumbai, India. He received the Young Investigator Award from the University of Vienna, Austria in October 2023. He has published his works in more than 30 peer-reviewed journals and 2 book chapters. He is a regular invited reviewer for journals such as Trends in Food Science and Technology, Communications Biology, Journal of Experimental Botany, the Plant Journal, Giga Science, Environmental and Experimental Botany, Functional and Integrative Genomics, Frontiers in Plant Sciences, PLOS One, and the Plant Genome, to name a few. We are highly honored and excited to receive him as a guest speaker on our Bioingene platform. So, Dr. Ghatok, we are all ready now. You can share your screen and we can start today's talk.
Can you see my screen now?
Yes.
You can see my pointer now over here?
Yes.
Okay. A very good morning from Vienna to all the viewers and the listeners of this webinar. First of all, I would like to thank the Bioingene, Dr. Subhabrata Mazumdar and Soma Roy for their kind invitation and introduction. I am doing my research at the University of Vienna Molecular and Systems Biology Lab, and our mass spectrometric facility is also called as Vienna Metabolomics Center in the University of Vienna. Today, I will be talking, as Soma already introduced, the Millets for Sustainable Future, a perspective on abiotic stress and BNI. You can also follow us on LinkedIn, Facebook, and also X to know more about our research activities and publication. Today, my talk is majorly focused on agriculture and climate change, and what is the history of the millets from where we have been evolved, and how the millets have been really going into Africa, then to India, and to the different parts of the world. I will be also discussing with you the important types and the health benefits of the different millets across the globe. What are the nutritional value of the millets in the world right now, and what we can really gain from the millets, and our body can really respond to the millets. We will be also focusing on that. And I will be also first studies of ours on the abiotic stress of pearl millet. And basically, we are focusing on the drought stress over here. So I will be focusing on the pearl millet drought stress and also the proteomics and the metabolomics sections of that. And next, we will be focusing on the biological nitrification inhibition in millets. This is a very important question to be answered right now. And I will be telling you more and more what is BNI and how we can really reduce the greenhouse gases which has been exposed to our environment daily. And at the end, I will be talking about our facilities and the equipment at the University of Vienna where we are working, and what are the different glass houses, greenhouses we have. And to end the talk, I will be also giving you some take-home messages to really understand what is going on from the whole millets research we are doing over here. Now, when we are talking about millets, first we have to know what is the agriculture and how is the climate change really focusing on this particular millet research. As we all know, the population is increasing, and it is predicted that it will surpass approximately 10 billion by 2100. And approximately in the first quarter of the 21st century, you can see we have already added 84% of the more population compared to the 19th and the 20th century. So the population is increasing exponentially, and now we have to really focus how we can really feed them. The world population is growing, but the arable land is not. So we can understand that the world population is increasing day by day and from year to year, but the arable land is really decreasing. And then in just 35 years, roughly, there will be approximately 2.6 billion people more on Earth, which will be directly depending on the agriculture. And on top of it, we have got 40% of the total food population productivity is lost due to the pests every year. For example, locust attacks, we all know in 2020, approximately 280 kg per capita food gets lost and wasted every year in Europe. And FAO, on the other hand, is estimating that agricultural production should increase 60%, and this agriculture has to undergo a significant transformation for higher yield, which can only be achieved through the technological advances. We divided the agricultural pressures on two different trends. Number one is the socio-economic trends, and number two is the environmental challenges. In the socio-economic trends, we have food, feed, fiber, energy, livelihood, and ecosystem services. And in the environmental challenges, we have got climate change, loss of biodiversity, land degradation, and water scarcity. Today, I will be focusing on the climate change and the food. Coming to the warnings of the climate change, we have seen there is a lot of warnings from the several years, but then also the most important warnings and the climate change which is affecting our livelihood are the melting of the glaciers, increasing the drought, increasing drought all over the world, marine ecosystems is getting hampered, carbon cycle is really going high, and there is also a fresh water shortages have been seen, and also there are many forest fires, food shortages, and the heat wave. They all respond to our agricultural productivity. And this will affect the food production and hamper our productivity for the future generations as well. Some predicted effects on the cereals by 2030, we have just listed it down. If at all you are talking about maize productions by 2030 will decrease, predicted to be decreased about 12%, and approximately there will be a rise in the 90% price increase in maize. If at all we are talking about rice, productivity may decrease to 23%, and there will be an 89% price hike. If wheat, 13% decrease and 75% price increase will be there. And in the other crops, we can see 8% decrease and 83% increase. These are all predictions, but then the predictions do come true if at all we are not taking appropriate steps right now. So where we have got so much of prediction happening, now let me give you a brief about what is the food pyramid doing. In nature, we have got approximately 30,000 edible plants available, out of which 7,000 plants that are cultivated for food all over the world traditionally, out of which 170 crops are grown commercial scale, and now this includes all the spices and the beverages, tea, coffee, etc. And only 30 crops that provide most of the calories and the nutrients, out of which only 3, wheat, rice, and maize, contributes and provides 40% of our daily intake. So you can see from this from 30,000, we have just 3 crops which has been giving us so much of calories. So we have to explore the unexplored part of the food pyramid. Coming to the big challenges from this as the increase of the climate destabilization, as I have already shown you, and also the crop yield are decreasing. So the food insecurity is increasing, hunger is increasing, and also the malnutrition is increasing day by day. Now, what are the major steps we can do? We can have and we can promote the underutilized crops, and also we can also do the climate resilient crops and increase their productions to sustain this harsh environmental condition. Now, first, I would really like to discuss with you how we can promote the underutilized crops from all this existence of the new crops and the most of the 3 major 40% calorie intake crops. Going to the history, so millets is one of the best solutions that we can have, that we can really get into from the wheat, rice, and maize. Other than these, millets is one of the best solution to really handle on this. So millets are the oldest farmed cereal grains in the world and domesticated in India and Africa, and this dates back more than 5,000 years ago. After the World War II, there is then corporations, governments wanted to increase the wheat and the rice yield and the market share by using the abundance of water, lots and lots of fertilizers and pesticides, and they called this as the green revolution. So we have already done the green revolutions to feed the population, but now the green revolution is putting a lot of hamper on the soil fertility and also the GHG gases. Thereafter, rice productions doubled, the wheat productions tripled, and this change dropped the millet shares in food grain production from 40 to 20%, leading to a serious agricultural environment and nutritional consequences. So the government policymakers had a very good chances to make this rice and wheat a staple crop for the world, and they were successful, and the millet production was really less from 40 to 20%. And 97% of the global millet productions and the seed demand comes from the developing countries such as Africa, India, and other parts of the world. And after being proposed and by India and supported by 72 countries to enlighten the world about the nutritional and the ecological benefits of the millets, the UN General Assembly declared 2023, as you all know, as the International Year of Millet, to recognize their importance in the global food supply. Now, coming to the types of the millets. So we all know there are different kinds of millets available in India and Africa. And the major millets are Johar, Bajra, and Ragi. They are also called as Johar is the sorghum, Bajra is a pearl millet, and Ragi is a finger millet. And the minor millets are foxtail millet, little millet, kodo millet, banyan millet, and corozo millet. These millets are divided in these two categories depending on the area of the production, the seed size, the seed yield, and the consumption pattern. And also the minor millets are been getting explored more and more, and they will also become major in some time from now on. Coming to the importance of the millets can be very useful for the planet from the farmer's perspective and from the human health. From the planet perspective, it grows faster, it's a shorter life cycle, put less stress on environment, grow organically, require very minimal amount of fertilizers and pesticides, and survive in a low water and nutrient content, and they need very less rainfall. From the farmer's perspective, it withstands harsh environments such as drought and dry heat, which is getting more and more throughout the world these days. Resistance to pests and diseases. They have got multiple uses for the food, feed, fodder, brewing, and also for biofuel productions. They can sustain the source of income for the small scale holder farmers. For human health perspective, it is very important to have it is very rich in the micronutrients such as iron, zinc, and calcium. It is highly in dietary fibers and antioxidant capacity. It has got low glycemic index and gluten-free. It is high in protein and the vitamin B content, and it is very rich in polyphenols and phytohormone content. Now, there are 17 Sustainable Developmental Goals adopted by the 193 member states in 2015. These are the goals which the United Nations wants to achieve by 2050 or 2100, out of which millet productions and marketing consumptions highlight 6 major goals. That is, the number 2 goal was the zero hunger, number 3, good health and well-being, number 8 is the decent work and the economic growth, number 12 is the responsible consumption and production, number 13 is the climate action, and number 15 is a life on land. To fulfill these SDGs goals of the millets are reintroduced, that is from the International Year of Millet, considering their nutritional importance to reach the zero hunger with lot of nutrition in them. Coming to the productions of the millet, and we all know that India is one of the major producer of the millets, which is approximately 38%. Not only production, but also India is a high consumers of the millet. And after this we have got Niger and the other countries, and also Mali, Ethiopia, China, Chad, Burkina Faso, Senegal, and Nigeria. They are all producing millet every day, and they are the major contributor of the productions throughout the world. And if at all we are coming to the Indian's pearl millet map, so you can see the pearl millet in India, sorghum is grown mainly in Maharashtra and Karnataka, Rajasthan and Tamil Nadu, And also, the pearl millet is mainly grown in Gujarat, Rajasthan, Maharashtra, Telangana, Karnataka, and Tamil Nadu. From sorghum, we have got around 4.24 million hectares of area which is growing sorghum and approximately 478 million tons productions every year. And from bajra, that is pearl millet, it is 7.75 million hectares area, and we have got and 10.86 million tons productions every year. Coming to the nutritional compositions of the millet versus the rest of the cereal, so you can see the three best caloric intake cereals, that is wheat, rice, and maize. They have got very low amount of all the macro and the micronutrients present in the seed, out of which rice contributes to the carbohydrate intakes of 78.2 grams per 100 grams of edible proteins, whereas in sorghum, you can see the highest amount of potassium content, that is approximately 328, which is very important for your body. Mg per 100 gram of the edible proteins. In pearl millet, you can see high in dietary fibers, iron, and also calcium. Finger millet is very good for the productions of the for very good intake, and they have contain a lot of potassium and also calcium. Finger millet is also called as the vegan milk. They consist of almost like 30% more or 30 times more milk producing, not milk producing, they have got the calcium producing capacity, and they can really help you to have this vegan milk, and also to have those who are lactose intolerant, they can have finger millet for their regular consumption. Oxtail millet is very good in protein and in fat contents, and also in the mineral contents. And if at all you are go porso millet is containing about 12.5% gram per 100 grams of edible proteins of the protein content, and also very high in the magnesium. So you can see the millets are full of nutrients and nutrient-rich components, and they will be really very helpful if at all people take them regularly instead of wheat, rice, and maize. Coming to the glycemic index. So what is glycemic index? Glycemic index in millets. It is a measure of how quickly a food can make your blood sugar glucose rise. Only foods that contain carbohydrates from the GI. So they are the foods which can really spike your blood glucose level. So they are divided into three, sorry. It is from low, intermediate, and high. In the low GI, you have got 1 to 55. In medium GI, you have got 56 to 69. And in high GI, all the components are from the 70 and plus. You can see all the low millet containing glycemic indexes are barnyard millet, porso millet, foxtail millet. Intermediates are finger millet and kodo millet and little millet. So if at all you are consuming it daily, it will also help you to lower your blood sugar level in the blood as well. Now, coming to my next point, that is the climate resilient crops, how we can really produce the climate resilient crop. To know this, we have to understand the role of the central dogma to identify the molecular markers. So at first, we have got the genomics, then the transcriptomics, then the proteomics, and then the metabolomics. And the transcription first, translation, and then the catalysis part of the metabolomics. So till today, in total, the genome sequencing are around 5,34,013 genome projects are already over or under construction, and they are increasing exponentially. But we have to really understand, do they provide about any kind of a trait information, such as the molecular or the physiological phenotype? Yes, but very less. In order to obtain the information, we need to decode the other levels of the central dogma, that is the proteins and the metabolites. Now, in 2020, we surveyed a report and saw only 30 to 40% of the phenotypic variance was observed from the genome to the annotations, and also hence we concluded that the rest of the 60% of the variation is not explained. And so only genomics will provide you around 36% of the phenotypic variance, unless you have to really go down to the different molecular levels. In order to understand this, we have introduced the genotype to phenotype relationships, and we are exploring them through our omics technology. And these all surveyed also shows that the GWAS analysis is only not enough to understand the phenotypic variance, and they have all rice, wheat, maize, sorghum, pearl millet, and everything was been really studied, and they almost contain 1000 genotypes, 500 genotypes, 300 genotypes, but they end up only with the 36% of the phenotypic variance. So we also need to understand how we can get to the phenotypic variance of 100%. Coming to my first study, that is the drought stress proteomics in pearl millet. So we have done the protein identification for the marker assisted breeding in pearl millet through the drought stress. And as I've already told you about the different millets, now I will be focusing on the pearl millet basically, because in the University of Vienna we are working on the pearl millet part. So pearl millet is commercially important because it is got the 90% of the nutrition 90% of the people are consuming it globally. It is almost 27 million hectares produced worldwide in 30 different countries. It is high in iron, zinc, and folate. It grows with very little amount, approximately 250 millimeter of water is required, and tolerates a very high temperature. Considering that wheat, rice, and maize can only sustain to around 32 to 40 degrees Celsius, whereas in pearl millet at 42 degrees Celsius also, the reproductive part are really fertile. Coming to the first important paper of the pearl millet, that was a genome sequencing, and this was done by the 63 scientists from 10 countries, decoded the pearl millet genome in 2017. From the University of Vienna, myself, Professor Wolfram Weckberg, and Dr. Palak Chaturvedi were also involved. We have decoded around 1,000 genotypes, which sequenced 38,000 genes, and there were also many genes which were also responsible for the biotic and the abiotic stresses. This project was led by Dr. Rajiv Varshne from Murdoch University. Coming to our first experiment at the University of Vienna, we have also done to a drought stress proteomics in pearl millet. Here you can see, to mimic the natural field conditions of the plants, we have transferred the plants in a high temperature polypropylene tubes, and we have imposed the drought stress by introducing these two sensors, one on the top and one in the bottom. And then we have measured the stomatal conductance from 10:30 in the morning to 6:30 in the morning next day for around several days. And then you can see it was around 10 to 30. It was from the 20 to 30 nanomole meter square per second of the stomatal conductance in the control, whereas in the stress, the stomatal conductance was decreased for around 10 to 20 nanomoles per meter square per second. And if at all you see the root morphology, in the control conditions, the roots were around 30 centimeter in growth, whereas in the drought stress, you can see it was deepening and deepening to fetch more and more water from the deeper layer of the soil, and it was around 60 centimeters, it went high. Coming to our all the data analysis which we did after the experiment. So we harvested the root, seeds, and leaves proteomics analysis, and from there we see from the principal component analysis, we can see the root tissue and the leaf tissue was completely showing a good variance, whereas the leaf tissue was not so hampered from the drought stress. And from the Venn analysis, you can see from the root stress 271 proteins, in the seed stress 159 proteins, and in the leaf stress 292 proteins were unique. So these proteins were subjected to a more deeper analysis, and we did tissue-specific drought responsive proteins, and this was done by the K-means cluster analysis, and proteins present in all three biological replicates was really taken in, and you can see tissue-specific in root control tissues, tissue-specific proteins in the leaf control tissues, and the tissue-specific seed tissues in the seed control tissues. And there are also stress proteins in the leaf, roots, and the seeds were also seen in the cluster analysis by the K-means. Coming to the identification of the drought responsive markers, so after combining all this analysis and also the K-means cluster, we come to the conclusions of the seed, root, and the leaves, and we have seen LIA, storage proteins, and the starch and the redox proteins was increased, whereas Kelvin cycle and phosphopentose phosphate pathways were really decreased. in the leaf, we have seen that the light reactions and nucleotide metabolism was increased, whereas the glycolysis and the other mechanisms were decreased. In the roots, we have got cell wall transport and the signaling proteins were increased, whereas storage proteins and the hormone metabolism were decreased. We also saw root to shoot crosstalk signaling proteins, which was increased in the GTP binding proteins, leucine rich transmembrane proteins were also observed. And from this, we have seen the regulatory network was also under construction from all the different millet related proteins, and we have jotted down in the subcellular level on the nucleus, chloroplast, and also in the mitochondrial analysis. And we also see what proteins were increased and decreased in the subcellular level of the pearl millet as well. Coming to our next study, that was the drought stress in C4 pearl millet and the C3 wheat. So we have seen the physiological proteomics analysis shows the superior drought stress in C4 pearl millet compared to wheat. Now for this, here we took two pearl millet lines from India, that is ICTP 8203 and 843. ICTP 8203 is a tolerant variety and 843 is the sensitive variety. Whereas in the wheat, we took the Iran variety from Middle East, from the middle part of the Iran, and this was TRI 5630. And in the wheat, we have taken the sensitive varieties from UK, and they were all subjected to the drought stress analysis, and we investigated the impact on the C3 and the C4 plant. So, coming to the physiological responses of the pearl millet and wheat, we have used these ODOM symbols. These are very unique symbols, and we have identified all the proteomics and the physiological responses, and we have plotted in these ODOM symbols finally. And here you can see in the wheat and the pearl millet, this is the well-watered conditions. The wheat root doesn't grow so deeper, whereas the pearl millet roots grow very deep inside. The root has the root conditions is also seen. The seed production is very maintained, whereas in the pearl millet, the seed production in the control conditions was fine, but then also it was lesser than the wheat production. But then when we subjected them to the white fife and to the 843, these are the sensitive varieties of the wheat and the pearl millet. When they were in the stress conditions, root systems went very inside the tubes, and then you can see they would have to maintain to fetch the water for the seed production. And also with the tolerant varieties, you can see there are many different varieties which were really going inside the soil systems of the tube, and they were really very having a maintenance of the seed systems in the soil. And also they were also going a lot of showing a less amount of the leaves. The leaves were really getting to a lot of stay green phenotype was not observed. So basically in the sensitive varieties of the pearl millet, and these systems were all we got all the physiological and the proteomics data, and we plotted in this ODOM symbols over here. Coming to the key aspect of this one, and we finally found out through the box plot the photosynthetic proteins, which we also saw it in the pearl millet and the wheat. And also we saw the carbonic anhydrase, Pepsi, and we saw the different regulations in the pearl millet and the wheat for the different part of the drought stress conditions in the photosynthetic activities. Coming to the regulations of the proteome, so we have seen the stay green phenotype, which is a major important phenotype in pearl millet. And stay green is a term that is used to describe the cultivars with the trait of maintaining their leaves for a longer period of time. With all the physiological and the proteomics measurement, we identified the stay green phenotype in the tolerant genotype of the pearl millet, where we found the photosynthesis proteins, photorespirations, 1433 proteins, and chlorophyll content was increased, whereas in the non-stay green phenotype, photosynthesis, photorespiration, and the chlorophyll content were decreased. Whereas in the stay green phenotype, ROS, oxidative stress, and the carbon and energy metabolism was decreased, whereas in the non-stay green phenotype, it was really increased. Coming to my last study of the whole analysis from the roots, exudations, and the BNI. So we have got the BNI, where we have collected the roots from and observed the BNI potential in the pearl millet, which can boost the sustainable agriculture goal. Here, I will be explaining you what is the nitrification and how we can stop the excess nitrification now on an urgent alarm, and how to control the GHG, that is the greenhouse gas emission from the soil, and below ground leaching of the nitrates, which is poisoning our fresh water, and how we can really control it. To feed the populations, we are extensively using the fertilizers. You can see when without the nitrogen fertilizers, how the populations have been really controlled, and how we can really feed the populations from this lesser amount of the nitrogen fertilizers. And when we are increasing the nitrogen fertilizers, we can feed around 7 billion people. So nitrogen fertilizers is very important. But this has an enormous impact on the negative impact on the atmosphere and to the livelihood. So we have used excessive chemical fertilizer synthesizers, which are nitrogen fertilizers. Nowadays, up to 150 Tg yearly is being used. A major problem here is that approximately only 30% end up in the nitrogen fertilizers in the crop plant. So climate change and even more current war conflicts which is going around the world is making even more vulnerable against this, which are not sustainable agricultural processes. An example, the price of the nitrogen fertilizers is not affordable anymore for the different farmers and the breeders in the world. Now, why we need the 70% of the nitrogen fertilizers escapes the environment? How we can really stop it? Because this 70% of the nitrogen fertilizers, when it is going to the environment, this is called a lot of soil degradations, and also the soil microorganisms are really helping the nitrification processes. And this soil cannot retain the ammonia and the nitrate, and they are leaching out to the soil into the groundwater and the environment, producing enormous environmental pollution. At the same time, nitrate-dependent denitrifiers producing large amount of the strongest greenhouse. This is the greenhouse gases, which is indeed a very are really getting nitrous oxide is getting outside of the agricultural soil management, which is exposing to around 78% of the contaminations in the atmosphere, and they are also having a lot of contributing to a global climate change as well. Food exudates are the natural occurring compounds, which are majorly the primary and the secondary metabolites. They are released from the roots of the plant in the nearby soil, which is the rhizosphere soil around here, and setting up a communications between the plant and the soil microbe. They are also a compound which exhibit the excesses of the nitrification, and the process is called as the biological nitrification inhibitions, or the BNI. So this is the BNI compounds from the root exudates, and we are trying to collect them for the better nitrification and to reduce the nitrification process in the soil itself. Now, for this, in the pearl millet, what we did, we grew the plants in the pot, and then we extracted the root exudates in water. And this is a schematic view, and then we concentrated them through the rotavapor, and then we subjected this root exudate to the UV HPLC, GCMS, and the LCMS analysis. In total, we identified 97 different compounds, which included tryptophan, synergic acid, T-coumaric acid, isoferrulic acid, and 2-flavonol. Determining maximum concentration in all the samples were also observed. In the GCMS analysis, we did 49 compounds were identified and quantified at a level 1, and these can be broadly classified into amino acid, sugars, organic acid, and other compounds. And for the LCMS, 32 MZ features for positive ion mode and 958 MZ features for negative ion modes by the in-house developed software MZ Fund were also identified. Going to the drought stress alterations of the exudation composition of the primary metabolite. So here you can see the different regulations in the pearl millet sensitive and also the tolerant genotype showing different regulations of the primary metabolite. And we have identified broad aspects of the primary metabolites, which are broadly classified into amino acids, sugars, organic acid, and other compounds. they have got a strong effect on the concentrations and the components of the exudates in the sensitive genotypes as compared to the tolerant genotype. Drought treatment enhance the concentration of the organic acid such as succinic, oxalic, lactic, fumaric, and malonic acid in both compared genotypes, that is ICTP 8203 and 84322B. Increased accumulation of this compound in the exudates of the drought stress plants can promote osmotic adjustment, which may help in maintaining root development to help access the water from the deeper layers of the soil. Now, coming to the biological nitrification inhibition in pearl millet, we subjected these root exudates to Nitrosomonas europaea. This is in AOB, ammonium oxidizing bacteria. And we see in the pearl millet stress treatment plants, the maximum total BNI was seen, and also the specific BNI in the stress treatment plant was high. So this also concludes a stress plant, that is 84322B, showed the highest activity of the BNI compounds. Now, to conclude all the results, we have made a conceptual model and a mechanistic model to describe the modifications and the key system parameters for the root exudations in the different genotypes of pearl millet of 84322B and ICTP 8203. Here, you can see in 84322B, due to the sensitivity, the roots were going deeper and deeper, and there were very high exudations, and the bacteria and the archaea were also having a lot of nitrification processes helping them, and then there were the BNI activity was reduced. Whereas in the ICTP 8203, it was increased because of the tolerant genotypes were not so deeper increased in the root exudations. Now, coming to the BNI compounds. So BNI compounds have been classified and they have been already identified in sorghum, Brachia humidicola, rice, and they are still finding in wheat and maize. So in sorghum, we have found out sorghalion, sorcatrine, and MHPP. These are the processes from the JIRCAS, that is Japan International Research Center. Brachia-lacton was found in seagrass, and in the rice, we have also found out 1,9-decanidol as one of the BNI compounds. And rest, we are still investigating and trying to analyze. From the BNI and from the nitrification processes in the root exudations of pearl millet, these are the two most important papers from our lab. And we have published this in Biology and Fertility of the Soils, and the Panomics platform in the root soil microbiome activity, and the BNI is published in the Trends. We have also seen a lot of proteins identifications from the drought stress in pearl millet, and we have compiled them in a survey. And this we have published in the Serial Crop Proteomics Systemic Analysis of Drought Stress. And this is published in Frontier. And you can have a look and you can see what are the different proteins from wheat, barley, rice, maize, sorghum, and millets. They are going and having a lot of significant changes and increases and the decreases in the different drought regime when they are applied to the different studies. Now, the major aspect of the millets for boosting the farm income in India is really getting a lot of advertisement, and people are really boosting the millet productions in India. Why this is happening right now? Because the ongoing International Year of Millet 2023, it has presents an opportunity to reap the benefits of the millet. And we are really exploring them with the potential to become a new staple crop other than rice, wheat, and maize. Most millets have a short growing seasons and can be cultivated in areas where other crops fail to grow. And the minor millets require more than 35 centimeters of the rainfall only. The absence and the intensifications of the millet farming lack the development of the genetic tools. So there are many different genetic tools, and the trait improvement tools are coming up, and we have to really explore it for the millet as well, which is not explored till now. People are really trying to explore in the different labs. And the challenges of the optimizations and the storage supply chains are limiting the crop productions right now, but we have to really increase them. A concrete roadmap is reduced to strengthen the small millet cultivations, and such as the denitrifying varieties and the identifying the suitable for the particular environment, and also targeting trait improvement using the genetics and the omics approaches. So we have to also see the central dogma of the millets and really try to improve them for the further modifications as well. And the progressive farmers, breeders, and entrepreneurs are the most capitalized on the incentives and provided by the state and the central governments in the country. So they should really capitalize them because central government is giving a lot of incentive for the production and also farming of the millet productions in India. Now, what are the ultimate aim, which is ongoing efforts on the climate change resilient crops? So whatever we have said till now, we are trying to adopt to the marker-assisted breeding and the marker-assisted selection of the genomic selections, which is only giving 60% of the phenotypic variance. And here you can see from the panomics, 40% of the phenotypic variance. And from the panomics and the environmental platform, we can get the rest of the 60%. Then we can do a correlated gene marker assistant studies, and this will identify for the functional validations and the population level increase. Then these particular proteins and the markers and the genes can be adopted for the gene editing techniques for the CRISPR-Cas and validations of the genes and the applications, and then this will be really helpful for the precision breeding. These are the new crop breeding pillars which we have identified. To this, we can also, after we have identified the genes, we can also do the speed breeding technique. So we all know what is speed breeding now. So in the conventional breeding techniques, the crossing and the inbreeding lines used to take around 3 to 7 years, but in the speed breeding pipeline, it has reduced to 1 to 2 years. Now, if at all we are doing this, we have to also identify the pipeline, which is the potential to double the rate of the genetic gains in the coming years will also be helpful to really go ahead and see what are the different genes which has been really important to sustain this harsh environmental condition. So this is the ultimate goal of all this effort which we are doing in the genomic selections, gene predictions, genomic studies, proteomic studies, and also the metabolomics analysis. Coming to some of our things which we have already published in the different journals. So here, Professor Wolfram Weckberg from the University of Vienna and Dr. Palak Chaturvedi, who is a group leader in the crops in a climate changing environment, they have edited two different special issues in the sorghum and pearl millet as climate resilient crops for food and nutritional security with other colleagues from ICRISAT, SEED, SEAT, and the CIMMYT, and have in the first one editorial, they have published 27 manuscripts, and in the second volume, they have seen eight manuscripts they have edited, and they have collected a huge amount of data and huge amount of research papers and review papers are also collected for the there are many papers which are very highly cited right now. So you can have a look in these particular editorials. Coming to our facilities and whatever we are doing in the Moses department. So we have got this Panomics platform in our department, and we have 3 generations of Orbitrap mass spectrometry, that is Orbitrap XL, Lead, and Q Executive. And for the metabolomic profiling, we have ICTSQMS, GCTOF-MS, and GCTSQMS. And this whole metabolomics platform is also known as Vienna Metabolomics Center. And this is headed by Professor Wolfram Weckberg in our department. Coming to the pearl millet, which is we are growing in Austria right now. So these are the pearl millet sensitive and the tolerant genotypes in our garden in the department of molecular. This was our old building, and this was the sorghum productions and also the pearl millet on the back. And you can see there are these small tubes which I have already shown, high temperature tubes, and the pearl millet is growing in the greenhouses of the University of Vienna. And these are on the fields of the lower Austria. So there is around 2.5 kilometers or 20 kilometers away from the University of Vienna, where we are going in the fields, and we are also growing this particular genotypes to fetch the seeds and to do the analysis. And these are our phytochambers and also the different analysis we are doing in the Austria. So from the glasshouse, phytochambers, and the environment, we have got 3 different glasshouses. That is tepidarium, which is having 16 to 20 degrees Celsius with a humidity of 50%. Caldarium, which is having 23 to 25 degrees Celsius and humidity of 55%. Iridium, which is around 26 to 30 degrees Celsius and humidity. This is actually the tropical glasshouse conditions where all the rainforest plants are really grown very good. And then we have the phytochamber. So these are around, we can grow approximately 480 plants of 1 kg pots in a soil in a large chambers, and they have got 3 chambers in our department, and we have 240 plants which can be grown in 1 kg plots in the medium chambers for all high throughput experiments, and they have got 3 small chambers and 3 big chambers, and we can grow all this in the controlled greenhouse conditions. And also there are different light intensities for measuring the different plants, for measuring and for the growing of the different plants. We have also got the convirants. So there are 6 convirants we have already using it for our different pilot experiments as well. Now, from all the experimental designs and the analysis, what are the different take-home messages I would like to suggest with you? We have extensively applied omics approaches to identify the potential biomarkers for support marker-assisted breeding, that is called as the smart breeding. This adds to a new concept of the fundamental understanding of the plant adaptation to changing environment conditions. And we start to understand the phenotypic plasticity that is in situ also. For the development of the sustainable agriculture, it is at most important to understand the molecular mechanism that I have already shown the central dogma and underlying the millet response to the abiotic stresses such as heat, cold, drought, etc. Now, when the enormous knowledge is gained from the omics data and the new gene editing technologies, we can identify more and more novel traits in millets and transfer them to the other cereal crops as well. And for the sustainable future agricultural breeding, we should not only satisfy the diverse human needs, we have to also focus on the nutritional aspect as well, but also adapt to the revolutions of the farming systems, which is technological driven right now. And we have to adapt to these technologies more and more to produce more nutritious food. Coming to my last slide, that is, I would like to acknowledge all my colleagues, Professor Wolfram Weppers, who is the head of the department, Dr. Palak Chaturvedi, Professor Gert Buckman, Krista Schlepper, Anka Belair, Dr. Marcus Tegi, Shuang Sang, and Iro Paredes, Dr. Laila, Lena Fragner, and Clara Premier. They are the main backbone of doing all these studies and all the experiments in our University of Vienna. Our two most very important gardeners to maintaining the pots and the plants in the greenhouse and the phytochambers, Andrea Stoppel and Thomas Joss. From Murdoch University, Dr. Rajiv Varshney and Dr. Vanika Garg. From Japan International Research Agricultural Society, it is Dr. G.V. Subarao. From the University of Ovidio, Dr. Associate Professor Luis Valador and Dr. Cristina Lopez. From NIB, Professor Christina Gruden and Dr. Ziva Ramsek. And all our funding agencies, FWF, that is the Austrian Science Fund, FFG, industrial fundings, and also Marie Curie and the ADAPT. And recently, we have also got a Grantham Foundation to work on the BNI activity. Thanks a lot to all the viewers who join us in this webinar. Now I am open to all your questions and your suggestions. Thanks a lot.
We will start with so we're collecting the questions from YouTube. The questions are coming, so we'll give them some time to put in all the questions and then we will question answers. So why not we start with the interaction, the interview section now. We'll start. Dr. Patak, our viewers would be highly interested to learn about your journey and how you built your career. Could you please share it with
them? Thank you so much for the question. I started my journey from the university, from the D.Y. Patil University, and I did all my bachelor's, master's, and PhD from there. During my PhD, I had a collaboration with the University of Vienna to measure some of my samples. And from there, we started with all the analysis of the pearl millet coming from the different parts of the India and the different part of the world. And then we tried to analyze them slowly and steadily, and then we accumulated all the research data. So the thing was that it was in during this whole process, it was my bachelor's and master's played a very important role to understand the research platform and the research idea, and then we really went ahead. I really went ahead to do the PhD, and this was the whole journey from there. And now I'm a researcher in the University of BNI.
And how was your experience in that from India to Vienna?
From India to Vienna, from the core education, from the basic education, and from the most important part, which were really important in the people to know and understand what is about crops and plant biotechnology, tissue culture. We have done it in our D.Y. Patil University, and that was really very important to understand all the fulfill knowledge from everything, and then that really translated to do a high-end research over University of Vienna. So we have got more and more facilities, and more people joined us and did a lot of jobs together.
What motivated you to focus on millets and abiotic strategy?
So when I started the millets during my PhD, it was really very helpful that this plant was not studied so well, and there was hardly any kind of data available. But when we started doing it more and more and went inside deeper and deeper of this particular plant, we gathered so much of knowledge. So this was the major motivation, that there is no data available. We had no reference papers also there. So this was the major drawback. But then when we went inside ahead and ahead, and we published so many papers and did so much of research, that was the major motivations to know particular drought tolerant, heat tolerant plant which is already available, and people are keen to know, and there is no information available. So that was the major motivations behind the study of the plants as well.
So how do you integrate proteomics into your research?
Sorry, Suma, your voice got cut. Sorry.
Okay, how are you integrating? How do you integrate proteomics into your research on millets and abiotic stress?
Okay, so the major thing is that with the proteomics data set, whenever it is very important that the genome is there, because the protein FASTA has to be generated. And if at all there is no genome, then we have to also see the whole family Solanaceae family or something like that. It is really very important to have it. And from there, we can find out from the UniProt ID what are the proteome level, what we have identified to match with the functional annotations as well. But other than that, the proteomics integrations has been really very when we started, it was not so easy, because the genomes had just come out, and then we have to integrate them with a different analysis part. But now it is much more easier to understand and to see what is really going on in the proteomics part, in the metabolomics part of the analysis section. So I would rather say that the proteomics journey was really very tough at the start, but after all, when we generated so much of data, it became really and really very helpful for the people as well.
So where do you see the future of millet research in the context of climate change and increasing stress conditions?
Thank you for asking this question, because this is one of the most important analysis and the most important part, that the millet is really getting more and more global view in the world right now. After this International Year of Millet, there were many people who were really interested, and the many scientists really are going ahead and doing a lot of work. There is a lot of work going on in India as well, because it is a hub of the millet, and then the millets are really producing a lot of more and more interest, and the markers which has been really produced from the drought stress and heat stress, and then the C4 to C3 translation, and the knowledge of transfer knowledge from the C4 plants to the C3, it is really going ahead. So they have got a huge market, huge research aspects, and people should focus on the millets right now to grow more and more, and to utilize this as an underutilized crop still now, and make it easier and available to the rest of the world.
So when talking about work and collaborating with build it more. So how do you approach collaborations with other scientists or research groups, particularly those with expertise outside your domain?
So majorly when we are doing any kind of a research, we see who are doing similar kind of a research, at least in the similar plants, but also in the similar concept or the physiological analysis. This is really very important because other than your own collaborators around your domain, you have to also see what are the different colleagues who are doing different kind of an work and who can really collaborate with us, what expertise we have got from them which we can include in our work as well. So we search them, then we do a communications with them, and then we find out that what if they are interested. We have to see their interest level as well, whether they are interested to collaborate with us, and then we try to merge the collaboration. So there are sometimes these collaborations are also financial collaborations, and sometimes the collaborative research projects are also going on. So there are two types of collaborations. From the collaborative research are much more because there are a lot of things to exchange between the two labs and between the two knowledge gained people, and rather than the other parts are also very important to really understand what is the basic goal of the whole project and the basic goal of the whole science behind the collaboration.
Okay. So, when we are talking about working and research and technologies, so there are many emerging technologies or methodologies nowadays. So, which one do you think, or which one do you think will significantly impact your research or this field, this growing field research?
So there are many technological developments are also going on, because the thing is that the uh, uh, from the aspects of the different technological developments throughout the world, like AI is coming out, drone systems are coming out from the physiological point of view, and also to measure all the physical data. People doesn't have to go to the field. They have got the different kind of an sensors available, which we can plot it on the field, and the data can be available from there. But the major aspect is the analysis of the data. So once we go to the different layers and deeper into the layers of the analysis, then only we can find out what are the statistical analysis is very significant, and it is really very important that we can really do more and more research and find out a complete negotiation panel where we can really see that what is really happening and how we can adapt from there with all the molecular data incorporated and integrated with the physiological data. So this is really coming up, but also with the technological advances, there is a very good issue, a very good or a bad problem is that there is a lot of data available, and we have to really scrutinize it at the different levels. So that is one of the major issues which is going on in the market of the crop research and everything.
So, so this is the challenge that you're talking about. So can you discuss a situation where you had to adapt your research approach due to unexpected challenges?
So it was during the COVID times only. So there were a lot of people who were inside the homes, and they were not coming out in the research fields and also in the labs. And then we have to really find out the people who can really go in and in the term in one term basis, like 1 hour or 2 hours, and then we can exchange them with the other people. Previously they were everybody working together, and we could do and we can do the levels of the all the analysis together at the same time. But now, and from the now from the COVID part itself till now, it is still going on that the different people are really hesitating to be in one particular room or one particular lab environment or one particular glass house. Many people, previously there were 70, 80 people coming together and helping us around with the different parts of the physiological data collections. But now we have got very restricted. One or two people can only go and do this analysis, and then we can do it in the time series manner. But these were the major challenges. We were not able to find out good. Shops were closed, the industries were closed, fertilizers, people to really help us in getting this particular seeds, the transfer agreements, so we had to really cope up with during the COVID time. But now people are really getting open up, and slowly and steadily it is getting better.
So, how do you stay updated with the latest advancements in proteomics and other relevant technology?
To know the advancement and to know all these things, you have got the social media. So the social media is one of the major platform right now. The Twitter, the LinkedIn, and everything. The the professional media. So whenever the paper is published, we see it over there, and we can see what are the different technological advancement and what are the different part of the proteomics analysis they have really implicated in that. So there are many in-house produced software and everything where they do the analysis for the proteomics analysis. We have to collaborate with them to know whether they can also analyze for our data as well, whatever we have found out. There are also many different proteomics techniques where we can really identify which are the peptide level identifications and tryptic peptide level identification, where we can really have the software's help, or else we have to also do it from the lab work itself. But if at all there is a software in situ, we can really go ahead and do all the other processes as well. So the major aspect is the thing is that we have to really know where the technological development is coming from and how we can really collaborate. So the updation really comes from the social media. The papers are available, and the new research aspects are there, so we can really see it, how it is going and how it is not going in the whole world scenario.
So we have a very diverse audience in Bioingene. So most you can say are PhD scholars. So can you give some tips on manuscript writing for this young audience?
Thank you so much for asking this question. This is a very important question to all the research scholars who are doing research. So the major aspect of the whole manuscript writing is on your results and the discussion part. So whenever you start a manuscript writing, always focus on the results and the discussions part, because that is the main base of your manuscript. And go into the data deeper and deeper, find out the meaningful identifications, meaningful things from your data. And whenever you are writing a manuscript, the results and the discussion part, you have to really analyze the data better, do a good visualization of the data, because visualization and all the figures which you make, that is the most important part. Because from there, if at all the visualization of the data is very good, it will be very easier to write the introduction material methods and the conclusion part. But once the data is visualized and you have got a good amount of hold on what do you want to convey through your manuscript, that is the most important part. We are reviewing a lot of papers from the different part of the world and from the different labs. So we have seen that the visualization and the data integration, data interpretation is not so much in depth rather than what should have been there in that particular manuscript. So put a lot of effort on the result and the discussion part, and then focus on the material methods and the introduction part, because your manuscript will be accepted only on your results and discussion part itself. And how do you make your figures? Because figures are the most visually challenging. And then when you are making a very good figures out of it from your results, it will be very good, easy to sail in a good journals as well. So I would suggest focus on our results much more than the rest of the part.
That's right. This session will end, this interview session is going to end with the last question, and then we will move on to the questions that you were collected. So what would your words of advice be to the students and scholars who are new to the field of plant science research?
So the thing is that now we are getting a lot of fundings. So the fundings in the crop sciences is increasing, but on the other hand, there are a lot of drawbacks also. War conflicts are also going on throughout the world. But the major important thing is that whenever you are getting focus on your basics like your bachelor's, your master's, your PhD. These are the basics of your all your work which you will be doing in your future. So whenever you are doing this work, focus on your work, do a lot of research studies, try to build your career from there, because there is a lot of data which you collect and then you do not have time to really focus on analyzing them, writing on them. Because if at all you are focusing on developing your ideas and your systems from there, from the bachelor and the masters, you can easily go up in your field or in your area of research very high. So it is from the fundamental part. You have to be really very clear and um, very focused, that you know that what you have to do. But you have to be really very explorative. You have to really see what all other people related to your research are also doing it, and how they are really analyzing, how they are really putting forward the stories. And they have to be really very helpful to each other and really getting a lot of data from the different parts of their data analysis, do the analysis, prepare themselves to really go up in the. And basically in the research, you have to really get into the mindset that you will be always getting rejections. Rejections is the one part and the parcels of the life. You will get manuscript rejections, offer rejections. You will not be getting too much of PhD offers at the start, but obviously when you are going ahead more and more you are applying, you will obviously get in more and more ideas. But then this will be really improving your process of getting higher and higher in your field.
So we will, I, Shubho, no, please.
Hello.
Hello.
Hi. Some great presentations. Slight us so delight. I not many people are uh, give the compliment for your presentations. I'm collecting many questions are there. Few are related to uh, millets, few are related to farmers' point of view, and some are country specific. So, as you see in your Zoom chat, I'm already pasted few of questions, but not all, and others are. I collect all the, but I'll share the others. So, different people are asking different questions. So, if we generalized, so there will be one thing is related to health benefit of the millets.
Yeah.
So people are asking for, is it helpful for diabetic? Is it helpful for pregnant um, females? Or uh, in point of the nutrition availability, that which millets or which type of millets is very nutritious. I'm sure you have covered the slides, but still for the sake of these people and for the question answer round, could you please repeat or answer these questions?
Yeah, from the diabetes point of view, if at all you are talking about, so the as I saw, so in the diabetes there are low glycemic index millets. So if at all you are using them in the daily diet, it will obviously decrease your blood glucose level in the period of time. So if at all you are doing it for 1 day or 2 day, obviously it will not have any impact. But if at all you are doing it for regularly for a period of time, for 2 weeks, 3 weeks, or for 2 months, 3 months, then it will obviously decrease your blood glucose level. But the thing is that you have to also consider what else you are really having food other than the millets. So how much oil, how much fats and everything. So these are also very intriguing factors that you have to be really very see that what is the millets is really doing from the diabetes point of view. From the pregnant woman's point of view, pearl millet is very good because it contains a lot of iron and zinc. And because of that, whenever a woman is pregnant, the first thing the doctors give is the folate medicine or the iron medicine to them, or the iron supplements to them. But if at all you are having a lot of pearl millet before and after your pregnancy, so it will also help you to retain the iron and the zinc content in your body. And this will really very helpful from this aspect as well. So these are the two different from the iron concept and from the diabetes concept. There are different millets which has been identified as the low glycemic index, intermediate, and the high glycemic index. And then you can take up with a different part of the different portion of that particular millet in your diet.
Hmm. And yes, and as you mentioned in your talk that it is more nutritious than the rice, wheat, maize. So the questions is Nahi is asking that can you really replace it? The millet can replace the rice, wheat, maize in the future.
Exactly. So the question is very interesting, but I would rather suggest that it will take time. It will take a lot of time, more and more years to come. But the rice, wheat, and maize, they were introduced after the Green Revolutions to feed the populations more and more. But previously, we were depending on millets and people were using millets as a staple food and the staple diet. Even in the even in the developing nations like Africa and different part of the world, they are having millets in a regular basis. So the basic important thing is that whenever you are doing this, and when the government policies are there, different political aspects are there, the fertilizers companies have to go down. So these are the really very political questions, and also there are many government policies are also there, how the country adapts to this particular millet acceptance in the market and in the society as well. So we can do it, but then there are a lot of ifs and buts before it.
Obviously, then it's not the full replacement. Maybe the
Yeah,
blending can possible. Like
exactly.
nowadays, the millet biscuits are available, millet
Yeah,
chocolates are available. So blending can initiate it very easily. But if
yeah,
maybe in future the proportion of the rice and wheat with the millet can increase. So
exactly.
yes, it is a futuristic views, and we are hopeful because it's and always taking a pill or some supplement. It's better to take a whole grain because it gives lots of other things with the fiber. So maybe millet can use in such a way that it can give the extra nutritions and fibers and all.
Exactly.
Uh, Shubhankar Mohanty, he asking for that whole genome sequence data. Uh, where it is it available publicly or where he can find that whole genome sequencing data of millets?
So till now, not all the millets are sequenced. I can talk about sorghum is sequenced, pearl millet is sequenced, finger millet is sequenced, foxtail millet is sequenced. But for the whole genome sequencing, they are available. But other than that, Corso millet, Barney millet, people are really putting efforts, and there are many labs which are really trying to accumulate that. But it will take some time. So whole genome sequencing are still going on, but for the major millets, they are available. So you can so they are majorly deposited in the NCBI or in
Hmm.
any kind of a public repository, and you can also contact to the corresponding authors of that particular manuscript who has published this particular paper, and they can also help you with the understanding and getting these whole genome sequencing data from them.
Hmm. Okay, so sequence is available and it can be achievable. So now that as you already present that you are working on abiotic stress like drought and salinity also comes hand to hand with the drought. So markers, they Joyce Sahu is asking for that what marker can we use to study drought tolerant in fox and millet?
So it depends on your tissue specificity as well. So
Mm,
if at all you are working with the seed, roots, or the leaf, so for the roots and everything, I would say there are hormone metabolism markers are very important. For the leaves, you can see the APA hormone metabolism is important. The carbohydrate metabolism is important, and also with the seeds part, you can see all the storage protein markers are very important. So it depends from the tissue to tissue, whichever you are doing it. But from the marker-assisted breeding techniques, if at all you are doing, then you have to also do a lot of integration pattern as well. So it is not only the proteomics, it's not only the genomics. You have to find out a marker from the all the analysis, and there are very few crops which has been really analyzed at the marker level, that is the QTL hotspot region or the particular drought responsive marker regions, and then from that region which particular marker is really helpful for your analysis will really depend on the how much depth the analysis has been done and what are the breeders are really looking for. So if at all the trait discovery is also very important, that marker is that particular marker is uh, defining which trait of that plant. If at all you want a longer leaves, if you want a shorter roots, if you want a shorter plant and more high yield productions, so that particular markers depend on the trait discovery as well. That these markers produces this trait, and then you can focus on that particular marker for your analysis as well.
mm. And must be there is a lot of publications on related to this type of
Yeah,
studies, and because it's a very specific and cultivar
yeah,
to cultivar maybe also it changes. So
different.
yes, so I will suggest you to search more publications on that. And just one question is came up when we are talking about the nutrition quality is uh, mm, Mangesh Pandit. He asked just we are taking is a this questions is a fun that millet does not good millet does not have good taste. Can you make it tasty? So
That's a very good question. So the thing is that I would suggest, and what we do is that you when you're having a millet flour with you, so mix it with your wheat flour and then you prepare your dough. So when you
Mm,
are preparing your dough, you have to really, so once you are mixing only 10%, 20%, 30%, then you are increasing your input diet. So you don't have to really start a millet diet or a millet testing start concrete, and the whole flour you have to take, you have to
mm,
mix it with the different millets, and then you can make it little bit tastier. And this is the one of the option. Other than that, you can have different kinds of commercial marketing which is already there. The millets, what Subo was telling about, on the biscuits are there, chocolates are there. You can start tasting them, and then you will have slowly, and your satiety value will also satisfy, and your taste buds will also really get increased towards this millet adaptation. So slowly you have to really mix your millets, you have to take a nutri cereals together, and then you can really eh, at the end you can really go ahead. And but there are many research going on this one to make it much more tastier and much more user friendly. But let's see how where we can reach in the future.
mm, yes. And taste also very personal issue. So
Yeah, exactly.
so maybe to target all the consumers with a similar taste, maybe not possible. But yes, blending is the good option to
Yeah.
dilute the taste.
And also, I would like to add over here that all the healthy food and healthy diets are non-tasty.
Obviously, and that's why the medicines are not.
Exactly.
uh, Then one question is related. It is very common questions to bioengine that people are always concerning about the gene genome editing and modifications and hybrid. So another questions must be from the another nations.
Yeah.
So the questions is that any side effect on, does it have any side effect on human being or on the gene sequencing preparation or modified or hybrid millets? So the question is the genome edited millets, if it produced and people are consuming it, then there will be any chance to harm their health.
Till now, we haven't seen anything like that, but then there will be a lot of millets in the coming years which will be gene edited and everything. But from the time of aspect right now, there are many millets which are already available commercially. So you don't have to really go for the gene editing right now because they are what gene you can only make a flavor gene editing or something like that to make it much more tasty. But other than that, they are drought tolerant, they are heat tolerant, they need very less water and everything. So you don't. So when the research will be there and the gene edited crops will be already be there, then we have to see all the preclinical trials of that particular crops, and then for how much it can retain and how much it can really produce a good effect in your body. Then only we can decide. But from the aspect at the present, there is no any kind of an problem with any crops till now. So let's see what the future holds on millets as well.
Mhm. Yes. Mm, one questions because three people are asking the same questions about the productivity of the millet.
Hmm.
They have a idea about the productions, and they mention it that it's a low yielding varieties. So
Yeah.
what is your take or what is your point of view on that, how they can increase the productivity or yield of the millets?
So the answer lies in the question only because then whenever there is rice, wheat, maize, they when they started to grow,
Hmm,
we applied lot of fertilizers on the soil. So and then they grow and the productivity increases. There is many high yielding varieties in rice, wheat, and maize. But if at all you want to grow millets to that particular level, so you have to have lot of other techniques to really have the high yielding variety. Obviously, the productivity from rice, wheat, and maize, the millets are very less. But then we have to really come to that particular level of growing it because now the world population is not concentrating on millets so much rather than on focusing on wheat varieties and everything. So once we have those varieties for the rest of the consumption pattern, then we can really grow ahead and go on. But
hmm,
till now, people are not so focused and the government policies are not making so much of efforts to really grow millets for the breeders and the farmers.
hmm. Yes, that's true. And few people asking for your email ID. I replied that informations of your social media, everything is will be in our website. And from Samir asking from Tunisia that are you willing to collaborate in a research project? I am working on palm millets in Tunisia.
Yeah, sure. Why not? It will be very great to collaborate. Let me send you all your project details and everything, what you want to do and what is your aim in the project, and then we can really focus on and really go ahead how to collaborate with you.
Hmm. Okay. And one mm, that the one general point of view, what I'm viewing, what I'm noticed in the most of the people mind, that the educating about millets to the farmers, because it should be something, maybe it's part of the government and their policies. It's not always the scientific approach from the science lab. It's
Yeah.
generally from society. So but we can talk about it, that people are concerned that how we educate to farmers to cultivate the millets, and what will be the benefit of the farmers if they are cultivating it. maybe because of the demand is not so high, or maybe the people are not taking as much they should take the millets. So
Yeah.
there will be a issue of cultivation, that why people are doing it. So what is your thought on that? Is it a
So
question? Hmm.
So I will just explain some things in the Vienna. So the images which I showed is from the farmer in Vienna. So when we started approaching him, so he was growing just maize and wheat in his farmland, and there were many other soybeans and everything which is widely grown in the European climatic conditions. When we started in 2015 and 16, the
Hmm.
millets were not grown in the field. So the climate was so dropping down instantly on some few occasions. So after we overlay one layer of the millets in the soil, and after 20, 30 days, it doesn't plantlate. And every time when we are overlaying again and again, the millets are not growing. But now the temperature conditions, whatever we said, that it is growing high and high. And
Hmm.
so this is the major aspect. So you have to tell them. So we ask for a small field of area in the agricultural field to the farmer, and he said that you can use this field and let's see how does it really growing. And we applied the sensitive varieties, the tolerant varieties to heat and the drought. And then we saw a lot of farmers really interested to grow millets. So these are the so first we have to really tell them that this will not hamper your soil, or your soil fertility will not get decreased. Farmers are very personal and very touchy with their soil, because there is a lot of bad examples where their soil fertility has really gone bad with the different GMOs applied over there. But now people are really getting educated, and farmers are really having a lot of collaborations and concerns with the researchers and the breeders. And then we tell them that once you are growing, we can also do some nitrogen fixing bacteria can also be there, so which can help them to grow very faster. And this will not hamper your soil system. But then obviously we have to tell them, we have to talk to them, we have to go and talk again and again to give them a small field. And then after growing them, after we build the confidence in them, then they will apply in their fields for a longer period of time. So building confidence in farmers is really very important, and uh, uh, first step to introducing this millets in different farmlands around the world.
Hmm. Exactly. uh, Someone just asks, is there any postdoc vacancy?
Sure, we can look into this aspect. You can just email me. We can have a personal
Hmm.
communications about this one.
Frantino Antoni asked that maybe he or she, I can't understand the gender, but obviously uh, we had achieved production of 1.6 ton per hectare of the millets. So I'm glad someone who is cultivating the millets is joined today's webinar. Yes, and
Yeah,
that is we are trying to build, that some
yeah,
platform where not only the scientists that talk about, should be the general people, farmers also show their concern, not only the appreciation, sometimes concern also. We entertain, and it's also motivated. We also think to thought, because I don't have any idea about the productions. So now I can see that 1.6 ton per hectare is achieved. So maybe
yeah.
it's not a not a low production. It's a good productions in terms of hectare. And okay, so I wanted to. These are the questions, and many things are coming. So we are at the end. So little bit, I have a personal questions, like when you choose a protein or targets from your proteomics data, then as you just a second, I just write something. List. Uh, uh, yes, BNI. When, as you mentioned, that many BNI compounds are isolated from millets, rice, maize, then my question is that how do you know that this is the BNI compound? So, is there any database, or how, or it need to be validated because it's a molecules, means something is related to molecules. So, what is the process to getting it and relate to the BNI? These things I wanted to know.
Okay. The thing is that when we are really focusing on the BNI activity and also with the major issues of the BNI compound, so when we are exudating the root exudates and then we are focusing on the isolations of that particular BNI through NMR structural predictions. So these are the
Okay.
NMR, nuclear magnetic resonance, and through the different processes when we are doing the GCMS or the LCMS analysis, we know at this particular retention index, at this particular MZ mass, this particular compound has been showing the peak. So when
Hmm.
we have got that identification, we know that this compound, and through the fractionation, we can take out that particular compound, isolate
Okay.
it, and then we go for the commercial, for the give the company for
Hmm.
the huge amount of productions of this particular compound. So this is
Okay.
for the NMR productions,
Hmm.
and from the NMR we isolate this compound, and
Hmm.
then when the company has got this particular compound, so what we do, we analyze them with the Nitrosomonas europaea, that is an AOB, that is ammonium oxidizing bacteria,
Okay.
and there are also few kits now available where we can really screen them how much with the normal. Whenever there is the farmers what they put DCD, nitraphyrin, these are the
Hmm.
chemical inhibitors, and when
Hmm.
we are doing to identify the biological inhibitors, in front of the chemical inhibitors, how much the biological inhibitors can inhibit that particular nitrification process.
Hmm.
So, like this, we have got this microtiter plate analysis where AOA and AOB, ammonium oxidizing archaea and ammonium oxidizing bacteria, they have
Hmm.
got a different strains. And then Nitrosomonas europaea and Nitrosomonas vienaceae, Nitrosospira vienaceae. And then we see how much inhibition has been there. And from there, we try, and this you can do for so many compounds together. So you can
Hmm.
do it, and you can just ask the company to make 1 mg, 2 mg, or 5 or 10 mg, and then you can see the inhibition, how much inhibition is being done with the DCD and nitraphyrin, the normal chemical compound, normal chemical fertilizers which are used in the fields with the biological inhibitors which we are there. And
Okay.
the best part of the biological inhibitors is that get degraded very easily rather than the chemical nitrifiers which stays in the soil, which pollutes the land, which pollutes the atmosphere. That is the
Hmm.
major issue.
Okay. And for the control purpose, you use the only the sand or soil, means for if the you use the control, because chemical fertilizer is available, so it's procured from the market. But as a control, which things is you prefer?
So from the BNI activity, it is not only available on the root exudate. So you can also find it in the soil system also. So
Hmm.
whenever there is a communication between the root and the microbes in the soil, there are a lot of chemical exchanges which is
Hmm.
already staying in the soil. So
Hmm.
you can identify BNI compounds also from the root exudates, and then you can see from the soil analysis what are the different particular metabolites are there which really shows the communication pattern. So this is
Hmm.
the major interest for the BNI activity researchers right now. Soil
Hmm.
and the root exudates both together.
Okay, because it's quite interesting for me because I'm a little bit aware about the BNF, biological nitrogen fixation, but
Yeah,
VIN is a little bit different and interesting. Means it is it's also important
yeah.
because not only the BNF, it's also important in to maintain the fertility and soil. Okay, now it's lot of appreciation and questions is coming and coming in. So let's end the session, and I will send you the rest of the questions or whatever.
Yeah, sure. You can send me an email and I will try to answer maximum of them. And then you can distribute to your webinar listeners, whoever wants to answer, wants to know the answer about these questions.
Right. Okay. So, okay. Then it's our end of the today's session, and Soma also came for the for staying the by,
Yeah, it
and
was so nice to see you, so nice to hear from you. It was lovely, and our audiences are really appreciating your presentation. Thank you.
Thank you
and
so much. Thank you so much, and I hope I did justice to the subject and the millets and everything.
yes, yes,
Yeah,
yes,
You were really wonderful.
yeah. Thank you so much.
yes, mm, uh, and you can see the justifications. People are asking for postdoc collaborations.
Yeah, yeah.
And obviously groups, as I know, and thanks for accepting our invitations. Maybe in the future we'll try to do some other thing in bioengine for the benefits of our young participants. Those are very energetic and asking many questions, and we should welcome them. And I try to answer their queries in mail and others mode of communications. So thanks one again for this lovely webinar. It's so informative, and it's called the end of international year of 2023.
Yeah.
So, but obviously, it's not the end of the millet. It's just a beginning.
Mhm.
And generally, this type of international years, particular any crop, it comes for the highlight the people about the importance. And we also try in Bioengine to make the highlight on the millets. And there are lots of type of millets and lots of variations, and it gives lots of research opportunity for the those are listening in this webinar. So there is an opportunity to work on millets. So thanks Annam for accepting the invitation.
Thank you so much. Thank you so much, Subho. Thanks a lot for the invitation and also allowing me to spare time with you and to show all my results over here. Thanks a lot.
Yes. It's a pleasure. We love it.
Yeah.
Bye. Bye.
Bye bye.